What is eDNA and how is it useful? In order to protect the living environment, we first need to understand which species are present and how their populations are faring. Identifying the many different species can be problematic, especially for those which are very similar to each other in appearance. Experts with the relevant ID skills are not always easy to get hold of, and you may need a different expert for every group of species you are looking at. There is not only difficulty in sourcing these experts, but also the time constraints of trying to identify hundreds if not thousands of organisms. One way to tackle this challenge and to speed up the process is to use the organism's genetic fingerprint, its DNA. Without even seeing a species, we can tell if it has been there by DNA left behind in the environment. These traces can be picked up by taking samples of soil, water or air. But firstly, what exactly is DNA? DNA is the blueprint of life. It is genetic material that codes for all proteins, functions and processes that make a living organism. DNA is found inside cells, either in the nucleus or in organelles such as mitochondria, which are responsible for producing the cell's energy, and chloroplasts, which use the sun's energy to produce food in plants. There may be hundreds of mitochondria or chloroplasts per cell. As we mentioned earlier, DNA can be picked up in the environment, meaning that one beaker of soil can contain genetic material from all the creatures which live in or pass through that area. Samples are taken back to the lab where DNA is extracted. In order to know what species are present, we must first sequence the DNA. Sequencing is the process we use to work out the order in which the 4 building blocks of DNA appear on the strand. An organism's DNA can be made up of billions of these building blocks. Instead of trying to look at the whole sequence, we use molecular barcodes, small fragments of DNA that are unique to each species, just like a barcode in a supermarket. The section of DNA that acts as the most effective barcode can differ between groups of organisms, so, depending on what is being looked for, it may be necessary to sequence a range of different barcode regions. The sequenced barcodes can then be compared to a database of all currently known DNA sequences, and the organisms can be identified. An essential step to developing a thorough understanding of the diverse communities of life will be to build a reference database of DNA barcodes for all of the flora and fauna on the planet. Even locally in the UK, this will take years, but is a work in progress. With continuing work, eDNA methods will fill in some all-important gaps in our understanding of biodiversity, yielding the potential to transform the science of nature conservation.